Hello everyone, in this video we are going to discuss various tests which are used to predict the specific groups of amino acids. So we'll be learning with some mnemonic here. First thing, that biuret test. The biuret test, the name itself telling this bi means two. So it is used to assess two peptide bonds. It is used to assess two peptide bonds to assess the two peptide bonds. Okay, ninhydrin test, please remember this ninhydrin test is used for detection of alpha amino acids. All alpha amino acids will give ninhydrin test positive. Okay, next coming into xanthoprotic reaction. Xanthoprotic reaction, you can remember xanthoprotic X. X means you can remember X, 6. So 6 sides are there, and 6 sides means it is the benzene ring. That means all the aromatic amino acids are going to give xanthoprotic reaction test positive. Okay, coming to Milan's test. Now if you carefully see Milan's test, NOL, in reverse direction it is NOL, so that is null. That means something equals to phenol. That means the amino acid which is having phenol group is what is going to give this Milan's test positive. Okay, so phenol group is present in which amino acid? Phenol group is present in tyrosine. Please remember the tyrosine is going to give this Milan's test positive. If you see another test, that is known as Follin's test. Follin's test is also, you can read Follin's test like phenol. Okay, it is like phenol. So pronunciation of Follin is like phenol. So it is also going to be positive in case of tyrosine. Okay. So this is also known as Follin-Shikhalchios test. Follin-Shikhalchios test. Next, coming to Sakaguchi's reaction. Sakaguchi is Gu. Gu, you can remember it as guanidine group. So the amino acid having guanidine group, that is arginine, is going to give positive test for Sakaguchi's reaction. Okay. Next is nitroprusside test. You can see so many S. Sulfur test itself is telling the amino acid should have the sulfur group. So these are going to be positive for sulfur containing amino acid, that is cysteine. Okay, so this is going to be positive by these tests are going to be positive by cysteine. Next, coming to Hopkins-Cole reaction. So you can remember it as Cole. So Hopkins-Cole reaction, or Cole, C-O-L-E, it is equivalent to Dole. Okay, D-O-L-E. So you can remember it as the amino acid having indole group. So the amino acid having indole group is something that is tryptophan. So tryptophan is having this indole group. Okay, so it is going to give positive result for Hopkins-Cole reaction. C is replaced by D, that is dole, that is indole group. Okay, next coming to Pauli's test. Pauli's test is, remember Pauli's test, amino acid mein pH is very important. Na? The one properties of amino acid mein, the pH, pK, isoelectric pH, those are very important properties. So you can remember it like Pauli's test ka P and H. So pH, P for Pauli's test, and H is histidine, very important, histidine. So histidine, that is having imidazole group. Okay, so these are the tests which are used to detect specific groups of specific amino acids.